A stem cell is an undifferentiated cell of a multicellular organism that is capable of creating an unlimited amount of cells of the same type, and from stem cells certain other cells can be created by differentiation. Meiosis is a type of cell division that results in four daughter cells, each with half the number of chromosomes of the parent cell, as in the production of gametes and plant spores. Embryonic stem cells are formed as a normal part of embryonic development. They can be isolated from an early embryo and grown in a dish.
Mesenchymal stem cells, also known as MSCs, are multipotent stromal cells that can be differentiated into many cell types. Here we have a drawing of a liver. The liver is the only organ in the human body that can regrow or regenerate. The liver is made of cells called hepatocytes. Hepato pretty much can be referred to anything with the liver. Hepatocytes. So that's what a liver is made of. So now we're going to go into how a liver can regrow itself. So if you lose this entire half of your liver, the part shaded in orange, if you lose this to some type of disease such as hepatitis A, B, or C, this part would have to be removed due to surgery. But the cool thing is the liver can regrow this entire part. It can regrow with no disease, nothing, in less than 2 weeks. And now we ask, how is this possible? So when the liver recognizes that they have a deficiency in hepatocytes, liver cells, the liver has stem cells which would pretty much become hepatocytes, and they'll come out of the G0 phase, which is a dormant stage, and they'll go into the G1 phase, which is pretty much heading into your mitosis cycle basis. Your prophase is when your nuclear membrane begins to disappear, which is being absorbed by your cell, and the chromosomes begin to condense. Yet you can't exactly say that they're separated because they're just trying to get organized, and they are at the center of the cells. And the centrioles move to the poles and produce fibers called spindle fibers. Metaphase is when the sister chromatids line up at the equator and they attach to the spindle fibers through the kinetochore. And each sister chromatid is attached to a certain spindle fiber, which are then attached to the centrioles. Then we have our anaphase. Anaphase is when the centrioles pull the sister chromatids apart from the nucleus, from the center, and they start pulling them to the poles. And then the centromere also is breaking in this stage. And then our last stage is telophase, and in combination with cytokinesis. So in telophase, your chromatids have reached your poles, and the cells begin to divide. Cytokinesis can also overlap with other forms of mitosis, and the nuclear membrane also begins to reform, and there's now two nuclei. In cytokinesis, animal cells and plant cells divide differently. In animal cells, they pinch from the outside called cleavage furrow, while in plants, they form a cell plate and it expands and then it separates. Now that scientists have at least understood how the liver, which I discussed earlier, how it regrows, now they've now they're starting to try and see how can we use how the liver regrows to regrow other organs in the human body, such as your heart or your brain. So there have been many different leads in how universities have approached this, but the biggest one, which most universities are holding in common, is the method through stem cells. Um, the um, stem cells are pretty much any cell which can differentiate into anything it really wants to be. So let's see. The stem cell in this case would pretty much turn into let's see, a neurocyte, which is a brain cell. It can turn into a cardiocyte, which is a heart cell, and that is etc. This list can go on forever because it can pretty much become any type of cell. So the thing here is, how can we put these stem cells into other organs? So first of all, we need to have a regulator which regulates and says that when we have a deficiency, specifically a um, deficiency of whatever type of cell, um, then let's see, we would have to create um slash produce new cells, and that's key here. Because if not, then they will remain in the G0 phase, and that is not something we want. We want them to go into that G1 phase. So if we can implant these new cells, if a cell remains in the G0 phase, then it won't exactly reproduce or go into mitosis. And that's a really huge issue here. As you can see, um, if it doesn't go into mitosis, then you can't produce new cells. And if you can't produce new cells, then there's no point in having the stem cells because it's going to remain the same. You won't be able to regrow the cells. Thus, the point here is when you insert them, and scientists are working towards it, when they put the new stem cells in, they should be able to identify and recognize deficiencies, as I mentioned before, and they should be producing new cells. Um, the specific stem cells that, in this case, they use is called embryonic stem cells, because it can be grown in many lab conditions, and another one is mesenchymal stem cell. Mesenchymal. The mesenchymal is the specific one which they typically intend to put in. Embryonic is more of a pre-stage of the mesenchymal stem cell. So what they try to do is they try to modify your embryonic stem cell to fit in with the body part you want it to be in, like the brain or the heart. And then it'll grow into a mesenchymal stem cell, in which when it's ready it can turn into these type of cells. And that's how scientists are going into regrowing new cells and organs. So this is a really interesting and a revolutionizing way of evolution and regrowing body parts.